How wide across, you mean, is our field of view?
Yeah.
That's a good question.
I was trying to look at the lasers and think it out, but my brain's tired.
I mean, it absolutely depends on how close we are to the seafloor. It's a pretty wide angle lens. If I go full zoom like this, I would say, I would guess it's the equivalent of about like a 14mm lens on a full frame camera sensor. That's a rough approximation. Yeah, that'd be a good Ed McNichol question. Our video lead on the
Look at this
Nautilus.
curly Q.
Yeah.
Could we get a zoom?
Yep, go ahead, video.
And I see the puffball at the top.
Something really small in there.
And there's certainly polyps at the top. I saw them sort of poking down.
Look
That's
at
so
all these
cool.
nice curls.
Looks like something out of Dr. Seuss.
Ooh,
Now,
nice flying,
correct
Mike.
me
Yeah, beautiful.
if I'm wrong, but this is
Wow,
counterclockwise.
Just
I was
pull
just
back a little bit.
thinking it looked like it changed direction in the middle of the stalk, but I don't
Frame
know
up
if
a bit
I'm
better.
just fabricating that because I
Ho-
want to see
hoping
that now.
for it,
Yeah. But there looks like some, and further down, some curls that are
Very nice shot.
beautiful.
Could we
Bridge
turn
nav.
lasers off for a second?
Thanks.
One more step, please.
Oh, yeah.
Oh, wow. That's gorgeous.
Can you get one with with a lot of negative space on the edge when
Coming
before
wider?
we pull out?
What a beautiful shot.
Beautiful.
Yeah. Thinking about future presentations. Thank you.
Yeah. Very cool.
And again, it's a
We're gonna lose it here.
Yep.
That was awesome. Thank you very much.
And I saw the shrimp in there.
Pull
a little
wide.
bigger one
Yeah.
for a bigger stock.
Those will be our fireworks for the 4th of July.
Yes.
You did see a shrimp?
I did see a shrimp.
Awesome.
Either that or see if they'll let us light off some flares.
I don't know about that.
Yeah, generally you don't do that.
So I bought all these Roman candles for nothing.
just put out security notice over the radio and say we're conducting training.
Did
Maybe.
you mark that as a highlight?
I did.
Awesome.
Or M, I'm typing it now. With a few seconds ago.
These are some nice low bait flows, just kind of creeping up towards the peak. So it appears like there's two kind of maybe volcanic conduits on this cone. This
So
is
is
the
that
second
two
one.
areas where the material is coming out?
Correct.
These little pockets in the flow.
Yeah.
Those are called kipukas.
Kapukas?
Kipuka.
Cool.
And what is
It's an area that the lava flow would have flown around something.
Oh, so like there was something there at some point that
Or it just didn't, it just kind of
yeah.
never went there for some reason. it kind of creeps and crawls
Mm-hmm.
down, you can kind of see how different lobes met up with each other.
Mm-hmm.
Could we get a zoom on one of the small yellow sea fans? There's one near the middle of the screen and one toward the edge.
Yeah.
On the right side. Either one.
Bridge nav. One more step, please.
Go ahead, video. You want the lasers back on?
Yes, please.
Wonder how long it took for that brittle star to find that coral down here.
Sorry about that.
Coming a little wider.
There we go. Punch in again if you
Back
want.
in.
All right, we're good here whenever. Thank you very much.
And there's a track sort of that I'm seeing on the rock as well. Would that be created by something like sea cucumbers as well? They kind of passed over them, but
I don't know. I didn't see it.
Yeah, I saw what you were talking about. I have no idea, but I saw it.
thank you.
Bridge nav. One more step, please.
So we can probably only squeeze about one more rock, correct? So in that case, I'll just wait till the next peak. Get one from each side.
Unless it's at the end of the dive, you could put it on the porch and put the port minute on top of it for recovery.
Okay.
There is a rock in the small
So
starboard aft compartment, so if you picked a really small one, we could probably throw it in there.
all right. Thank you. Keep that in mind.
About 15 or 20 meters to the peak here. Looks like we're just about looking at it.
Cool.
Very cool.
Ken, you can check out in Argus View to see a kind of larger context. And in Argus View, I also think I see some sponges.
Yeah,
To the
straight
left?
ahead.
Yeah,
I thought they
straight
looked
ahead
like, I
now.
think it's just
Oh.
sediment.
Oh, sediment.
Dang.
Yeah,
Yeah, I
that's
thought
kind of
so
trick your
too.
eye.
From
doesn't it?
above, they really did look like some vase-shaped ones.
Very convincing.
Yeah.
Alright, start stepping our way down slope and over to the next cone.
Yep.
Bridge nav.
And a
Are
note
you wanting
about-
to see any more of the slope
Request
of this
20
feature?
meter step.
Okay.
Bearing 135.
Are we gonna fly off bottom down this
We're
one?
gonna have to. It's really steep coming off, so.
You want to go off bottom?
Well,
If you want to take the time, we can spin around and back down it. Or if we do what we did before, we could do 0.5, right?
Yeah, we could track a line over.
I think we can do that. Don't you think? Do you want to see the slope? Yeah, let's go straight.
All right, roger that.
Let's see another one.
Straight into abyss.
And a note about those brittle stars that we were seeing, they are kind of going up into the water higher up
Bridge
so
nav.
they can grab onto that marine snow.
Request we track a line bearing 130 at 0.5 knots.
This next cone looks
Thank you.
perfectly symmetrical.
Yeah.
Crazy.
I know I was sleeping during Rob's watch, but do we know if Dr. Rob McColney got any better ideas about the formation of the moats at the base of these cones from these visual dives?
I haven't talked to him about it
Yeah.
since the dive started, but he'll be on the next watch, so we can ask him at the watch change.
Yeah, at the watch
Yeah.
change, he was excited about being able to record the Whipple orientations, but other than that, I don't have any information.
Gotcha. So stay tuned, viewers. Rob will be back in the seat, and you can hear from him.
Kind of a bluish call or a sea fan. And a fish. Maybe a
Nice.
halashore.
I see the fish this time.
Where's the fish?
To the left. It's about to go off
Oh,
screen.
I saw it for a
Oh,
second. Okay,
There
phew.
it is.
Oh, it's following us. Thanks,
It's
fish.
putting on a show for you.
Okay.
Yeah. Picking up speed on our line.
How many knots will we be doing now?
We will be doing 0.5 knots.
Were we doing 0.3 before?
Yeah, so for the other downslope flat area or for across the top?
Across the top.
We were doing 0.2, 0.3.
Oh, 0.2. Okay.
Depending. All depends on momentum and how often I call on the steps.
Gotcha. Thank you.
The moves we're about to be making, we're talking about how we are currently at the top of one of these volcanic cones. And if you look in the quad view, you can see that we're on the top of one where you can see our position in the top left corner of that bottom left screen in the cloud view. And so we're going to be kind of taking off into the water a little bit to scoot across to this next cone that you can see in the bottom of that
Go
bottom
ahead and reset DVL.
left quad view.
Well said.
Thank you.
We're going to lose it again in a second anyways, but... All right. Slowly but surely.
And so remember, you can write in your questions to nautiluslive.org. If you look at the bar right below the video feed, send in your questions about what we're exploring, about some of the things we've seen, about where we're going, life on board. We'll do our best to answer that as we explore here off of Palmyra Atoll in the Pacific Remote Islands Marine National Monument. This is our second dive of this leg of the expedition. The first dive, we took a look at an area that we refer to as the Kingman Ridge off of Kingman Reef. And then we transited down a bit towards Palmyra Atoll. And we're now southeast of Palmyra Atoll looking at these volcanic cones and the moats associated with them.
You just push in past that schnozzle there video. Thank you.
And we've still got a bit of time left on this dive. We started last night, right? We started this one last night?
About 4 o'clock
Okay.
yesterday afternoon.
It's blending together. Started last night, and we'll be probably recovering sometime this evening before we head to our third dive site of this expedition. But you still got quite a bit of time to hang out on the seafloor with us as we explore these volcanic cones. We've been sampling the biology of the area, lots of corals that are of interest to Steve Oskovich, our lead biologist here on board, deep sea coral biologist.
Yeah.
And we've also been picking up some rock samples of interest to Dr. Rob Picolny, and we've got one rock on this picked up on this leg, right? Only one, or did we get another one?
Just one for this
Just one.
watch, at least.
Just one.
But
It was
five,
a good one, though.
five total for this dive.
And we can have 3 more?
I can, but I would have to crush all of Steve's biology samples, so
Got it.
I'm going to try to squeeze maybe one more,
Yeah,
maybe 2 if we can.
one or
we'll
2
see.
more.
Thought you were going to say, and so that's what I'm going to do.
We're just going to reach in there and just pick up those things and eat them out.
Yeet.
Toss them downhill.
That's
Video
some
from later,
biology.
just taking them all out, throwing them away.
And then I'll spend the rest of my time in the brig.
Just beat it all up, trying to take them out and put them back on top of the rocks.
Oh my gosh. Jamie's probably not having fun with this conversation.
I am not taking it seriously, so I'm enjoying it just fine.
I think she got excited about the idea of a brig on Nautilus.
Yeah, I think I did start grinning at that point.
So as we move to this next cone, are we going to be doing what we did before, kind of moving through the water off of the bottom?
Yeah, that's the plan.
Kind of booking it over to our next spot?
Yeah. We shouldn't really hit any sediment plains on the bottom here. I'm sure it will kind of look like it does now, just kind of rocky with some sediment. But then imagine a steep slope, lots of volcanics.
And thank you to everybody joining us, watching on this dive. We got quite a international audience this morning. Got lots of friends in the United States, the UK, Canada, Spain, Germany, Austria, Turkey, Sweden, Puerto Rico, Norway, Netherlands, Japan, Israel, Hungary, Finland, Estonia, Brazil, and Australia.
Wow.
Woohoo.
Wow.
Wow, nice.
Global audience.
Quite
I
the
like it.
smattering.
Thank you for watching.
Smattering. I like. I'm gonna add that to my list of descriptive things for like variety.
Just a, a smatter here, a smatter there, a smattering.
I always use various,
Oh, yeah.
and that's like so boring compared to smattering. What is this? Like a cornucopia, a smattering of
A
things.
myriad.
A myriad.
Plethora.
Oh. It is vocab
Go
hour.
play the synonym game.
Our audience really enjoyed us using that term yeet here. Said, oh, the times they are changing.
Trying to be hip to the kids.
We're doing great, Kevin. We're doing great. When I'm at work, kids ask me to dab and do all sorts of things on stage, and I refuse. But I did get compared to a Fortnite streamer recently with my blue hair, so I guess I'm cool.
You got compared to a what?
A Fortnite streamer?
Oh. I was like, what?
Ninja?
A what?
It-
No, I think I know what that is.
I'm guessing they're talking about Ninja.
Yeah.
Yeah.
Kid came up to me and said, Ninja. And I said, what? And he said, Fortnite. And then he flossed, and I said, oh, okay.
Nice. Am I the youngest person in here?
I would imagine.
21?
Yeah, I'm
Oh, yeah.
26.
Yeah.
Who's the oldest?
We don't have to have that.
Yeah,
conversation.
that's a game we can avoid.
Who's the wisest?
It's about life experience, not age. Alright, DVL seems to be going on. I'm going to switch over to USBL.
Roger.
And we were talking before about dormant versus active areas, like these volcanic areas. And we were saying that evidence of activity would be things like seeps in the area. Was that correct?
Venting.
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 Okay.
Any active kind of gases coming out.
Yeah. We've got some viewers wondering if there's been any evidence of anything like methane seeps in these areas.
Not that I've seen, but doesn't mean there aren't. But typically you find methane seeps more on continental margins where you have high sediment input. I don't believe you find them on geos and
Yeah.
ancient terrains.
Yeah, we've explored, or Nautilus has explored a lot of methane seeps along the west coast of the U.S., especially last year, the Cascadia Margin cruise. So if you look in the photos and videos tab on nautiluslive.org and go back a little bit to some of the highlights from last season, there's, I think, a couple entries about the methane seeps along that margin.
Yeah, methane clathrates, what they're called, require a very specific kind of temperature and pressure gradient. So that we're pretty deep here, probably quite a bit deeper than a lot of the methane seeps are found along the continental margin.
Mm-hmm.
So more on the continental margin and Arctic permafrost. When we were in the Caribbean, we did find a what looked like a seep site off of a volcano, but it was created from a big sediment slump at about 3 kilometers down the slope.
Sounds cool.
Yeah.
And if you're wondering about the makeup of the crew of Corps of Exploration here on board Nautilus, we have our team, our ship crew that takes care of making sure that the ship is working and the engine's running and keeps us safe. We've got our captain and our first, second, and third mates who are also, if anything were to happen medical-wise, they're there to help us out in that regard. And then you have your science team.
Come down a little bit.
Sure.
Pilots, video engineers, mappers, navigators, all the scientists that are, planning these dives, and science communication fellows like me.
About 100 meters to this intermediate area down here.
Roger.
About how long till we reach that other cone, Chris?
To the peak. Give me a second.
It's going to be exactly at the watch change.
I was just thinking
right.
that.
I think we should get there before. So,
To the peak?
or yeah, probably right at. Never mind.
That's all right.
We'll just speed it up to 2 knots and just truck it over there.
Let's preserve the cable, please.
Let's see. So yeah, we're looking at exactly at watch change.
My delta's high, but I'm
Yeah.
gonna bridge it by like 5 meters.
Actually, probably for peak a little after, just because we'll slow down coming up slope.
Makes sense.
But we will reach the other one and start to go up it on our watch.
That's good.
For a minute or two.
For yeah. Hey, whatever we can get.
Exactly. I'm excited for recovery and seeing those samples. Fish. To the left, to the left. So the debris that you're seeing, again, that's the what we call marine snow. So that's what's forming the sediment down on the bottom. And it's debris from the upper levels in the water column that are falling down. So that's things like, when animals in the upper levels are eating things and sort of their crumbs, for lack of a better word, as they fall through the water. Fish scales, poop, slime,
Mucus.
all sorts of stuff.
I guess mucus and slime could be the same thing.
Speaking of slime, do you all know that the slime from a snail can actually be considered a liquid crystal? Because based on the pressure that the foot of the snail is putting down onto it, it can be stickier or allow them to slide smoother across the surface. Just, a little snail fact.
How is that crystal-like?
Liquid crystal?
Liquid crystal?
So it has to do with the molecular structure of the slime and the way that it interacts with each other. And so the shapes of those, I don't know exactly how to explain it, but things like color changing, like due to, those liquid crystal papers where the color changes based on heat, like mood ring stuff.
Mm-hmm.
That's also a liquid crystal. And so the heat is changing the orientation of those of the pieces that make up the liquid crystal, and so the way that they're oriented changes the light that they're reflecting.
Interesting.
Yeah.
And go ahead and put us back on DVL.
Roger.
Reset.
And we've got some viewers who are really interested in seeing the wet lab view from the last time when we did the collection or the sample processing. Ooh.
Zoom in there a little bit. Never mind. It's coming. Oh.
Beautiful.
Lobate ctenophore.
Mm-hmm.
Bad spores.
Hitching red.
Oh. But what I was saying before the lobate ctenophore, almost forgot that I was in the middle of something.
Squirrel.
Our viewers are saying that they enjoy seeing the wet lab view and seeing the sample processing, and they're wondering if there's any plans in the future to give more camera options during that period. I guess they want to see up close and personal, see Jamie in action.
Not at the moment. We don't have more cameras planned, but stay tuned.
You do a head strap GoPro.
That would be cool. Now, for folks that have done a lot of dives like this, do we normally see marine snow pretty uniform across different areas at these depths? It does seem to be sort of a uniform amount that's falling.
I think it does vary location to location. I mean, it's
I guess it would also
effectively the
depend on
sedimentation rate.
currents and stuff coming through.
Is there a average rate at which it falls?
In the global ocean?
Yes.
Probably.
Because I'm just thinking, it's obviously very slow, and we are 1,650 meters down.
Yeah.
So the age of this could actually be pretty old, right?
Mm-hmm. It's coming from all levels. according to knowledge I am currently gaining about marine snow, says that it's not uniformly distributed in either space or time, but it is usually found in higher concentrations in the upper water column.
Looks like we're reaching the end of our rocks.
Yeah, we're just about down at the bottom. Headed across a little flat rise here.
So Al, deep sea sediments typically have sedimentation rates less than 30 meters per 1 million years. And rates
What?
as low as 0.1 meters per million years have been reported.
Wow, so we're talking, this area we were saying was 500 meters of sediment?
No, upwards of, well, right where we are right now, maybe 500, but
The area in
the
general?
area in general is between 800 and I think 1,500 meters.
So if that's 30 meters a million years, and
Actually, I can just tell you the sedimentation rate for this area in the last, 2 million years is about 1 to 2 centimeter per 1,000 years.
all right.
Many millions of years.
Yes. Old debris. And we got a question about the wet lab. Jamie, you back there?
I am back here.
We've got a viewer wondering about a device with a small plastic handle that you often see the scientists pumping. Is that some sort of pipette that you are all using in the lab?
We almost never use a pipette these
Okay.
days.
eDNA maybe?
eDNA doesn't really have a white plastic handle, and we don't really pump anything. We do have a squeeze bottle that we get ethanol out of.
That
That's
might be
sort
it.
of a clearish white thing.
Oh yeah.
Yeah,
So
but
that's my best guess. Ethanol
do
bottle.
those bottles have the curved tubes to deposit the ethanol?
Yeah. Has like a spout out
Yeah.
the top.
Might be that, viewer.
If that doesn't sound right, give us more information and we will try to figure it out.
Yeah.
When you guys take water samples for eDNA, with the marine snow in the sample, has the DNA already degraded by the time it gets down here, or do you get DNA samples of like surface life?
I'm not sure. I haven't been involved with any of the processing. I got the impression that they mostly were able to tell about deep water species that were, within some close proximity of where we took the water samples. So they're probably getting little signal from surface waters, but I don't know the extent of degradation between when it was on the surface and when it gets down to these depths.
Gotcha.
That's a good question, though.
Is that anything, or is that just like scat? Video?
Let's zoom in.
I think it might be a little coral or something.
Yeah.
Good eye, Kylie.
Well, trying to find us something to look at.
Maybe a sea pen.
Yeah.
Yeah. Different from the sea pen we saw earlier that had the long stalk, but there are sea pens that have
wide?
more of this structure as well.
Okay.
Shout out to my other snail lover writing in.
So it'll respond faster now.
Snails are great.
Okay. Okay.
A little
Okay.
bit more about marine snow. So we were talking about sedimentation rates, but there are also sinking rates. So marine snow that starts at the top can sink about 68 meters per day. I'm looking at a study that was off Monterey Bay, California, and they saw rates of 43 to 95 meters per day.
Where you think it's going to be, because if you do get a good shot, you really don't
So
want that.
gives you an idea of
Right.
how long it would take something that originated in the surface to make it all the way down to the seafloor.
And I've also got, it looks like a little message from one of our scientists ashore, Thomas Gilderson, who says that recently exported fixed particulate organic carbon sinks pretty fast. I do not 100% know what the beginning of that means, the recently exported. Does that mean debris? Science team, help me out here.
I think that just means, like, let's say someone pooped.
Got
Yeah.
it.
Yeah.
That-
Or
we can say that.
a skin cell got shed, or some
So like
music
when it
was expelled.
leaves the
leaves
organism.
the organism, yeah.
Got it.
Yeah.
Thank you, Thomas
Mhm.
Gilderson. And thank you, Science Row.
Were you seeing that on the science chat?
Yes, it was a
that's
private
weird.
message to me.
Oh, okay. Because I was going to say it didn't pop up on.
Yeah, I didn't realize because I didn't realize you could get little separate tabs
Oh,
there.
Yeah. Thought something was wrong back here.
No, you're good. And on the quad view, you can see the HerCam, the Argus cam, and again that map that Chris, our navigator, is looking at. And just to confirm again, the position of the ship, it is scooting sideways right now to
Yes, we
get our
are.
position. Yeah. So yes, viewer, it is moving sideways.
So the ship itself, the bow is pointed southwest,
Video
but
zoom?
we are going southeast.
Another one.
The lobe 1804.
Yep.
And come wide.
Oh my.
You're getting good at those zoom positions.
Thank you.
You set your shot up,
Mhm.
and it makes your life a lot easier.
Mhm. Okay. Yes.
Getting a return on
Oops.
sonar about 30 meters out.
What'd
Oh,
you say?
never mind, we were just close to the bottom. Oh no.
All right.
So now we're in an area between these volcanic cones, and so we're seeing more sedimentation, possibly something over here.
Video zoom.
Maybe another type of sea pen, or
It
is
looks
that a
like
coral?
it might be on a small rock, so Maybe it's a coral. Could we get any tighter?
Coming in.
Doesn't
Full
seem to
zoom.
have polyps. Well, no.
I don't think it does.
No polyps, so it's not a coral.
And
Sea pin, probably.
gotta go. Come wide,
Yep,
please.
thank you.
Coming wide.
It's getting better.
Yeah, Kylie.
Thanks, guys. I appreciate the support.
You're awesome.
I like the YouTube thing. I'm gonna go back and watch.
Yes.
Take notes.
That's a good idea. We're seeing a bit more of these. Are these just pieces of basalt, or would these be nodules?
Probably pieces of basalt. I don't think.
Too angular.
Yeah, the too angular to be nodules. I don't know what nodules here would actually
Video zoom.
start on
Got it.
and nucleate off of.
Pop quiz, what is it?
Stocked hydroid.
Hydroid.
Yeah. You guys are good.
It's a call.
Not this time.
Looks like it's got that little thing coming out the middle that we observed on the other one too.
Yep.
Sort of a different color.
We need to look at stocked hydroid anatomy, figure out what that is.
And come wide, please.
It's like something Georgia O'Keeffe would have painted.
Yeah.
What are you?
Looks like another sea pen, maybe.
Got about 100 meters. I'll check our swing.
Okay, sounds good. Can't stay here too long. I'm gonna punch out ahead.
Jamie, we've got more details on the mystery device in the wet lab.
Oh, good.
They said the squeeze bottle is not what they meant, but they
Okay.
were always curious about the squeeze bottles,
Okay.
so thank you. They were saying that what they're talking about was a 1.5 foot tall device, metal and black, that sat on the
tongs?
bench, had a little tube coming out of it, and where that tube went in, there was what looked like a white handle, but it looked like it was being used to ratchet something.
Let me think on that for a minute.
Was it on the Gorda Ridge cruise, or is this something
Oh.
that's regularly used?
Yeah, if it was a previous cruise, it could have been a science-specific
Yeah.
that a group of scientists brought on that we don't normally have. So yeah, let's wait and hear back about that.
Yeah. So let
Was
us
it on
know.
this cruise or was it a former cruise?
20 questions.
To
getting
be
closer.
continued.
Are we sure it's not the metal tongs?
The
Long tongs.
long tongs, palm fronds.
And we're starting to see some marks again.
Video zoom. Let's see.
Does look like another sea pin.
And so how do things like these sea pens, How are they
Come wide.
in the sediment? Do they have sort of a root kind of shaped thing that holds them in there?
Yeah, there are different ones. Some prefer growing on hard sediment, some on soft, but the ones that are on soft sediment do just sort of stick part of their stalk down there to hold them in place.
So they just settle out of the water column when they're
Yeah.
young and
Larval form would float down, pick a spot, and start growing.
And as we approach the end of this watch, I am going to have to scoot out a little early for interaction. So I'm going to have to turn the question button off for a little while, but no worries. To our audience, we will be back later for most of the rest of this dive to answer your questions. So we'll be
Al,
back.
if you want to leave it up, actually, we can monitor it from back here.
Oh, you want to?
Yeah, we're happy
Oh,
to.
awesome. Well, in that case, you can still send in your questions. And hopefully more details about that mysterious lab object. All right. I'm going to scoot out right now. We're going to try out a mobile interaction. And I'll see you all later.
Have fun.
Good luck.
Yep, that's what I thought. So the mysterious instrument was from Gorda Ridge. It was a guest scientist using a specialized tool.
So unfortunately, that means I don't know what it was used for. But it was fun trying to figure it out, so thank you.
Name that instrument.
Oh, before I really go, we did just get a message in from our friend. It was on Gorda Ridge. And they think it was Sean Silva who was using the device. And don't apologize for the goose chase. That was fun. All right, bye for real. Talk to you later. Howdy.
Bye, Al.
What was the description of the instrument again?
Let's see. 1.5 foot tall, metal and black. On the bench had a little tube coming out of it, and where the tube went in, there was a white handle of some sort that was a pump or ratchet of some sort.
Video zoom.
Because I don't know if I'm 100% right, but I swear I was on the Nautilus Live Instagram, and there was a large black tube that was on there, and I think the description was for like for the water samples, keeping them pressurized so gases don't bubble off.
Yep.
That's the closest thing I can think of that description, but
Take a look at the Instagram.
Yeah, nice flying.
I think it was on one of the pinned science stories, possibly.
Yes.
Next step will have to be manipulation work. It's like maybe when we're on a blue water watch, you know.
Yeah, if we come up, I'll definitely use it. It's there for that we don't have one set up in the shop.
I know, that'd be cool. Something to think about.
Alright, I'm going to hold position and check our swing. Bridge nav. Request hold position. So as we swing over, we should be right on the edge starting to go up.
Sounds good.
Did they ask for what? Oh, sorry.
I feel like we just keep setting Rob up perfectly for his watches.
Exactly.
It really is a value service.
And I bet as soon as I go take a nap, they're going to find so much
Videos in?
biology.
Coral garden.
Yeah.
Ship was sliding a little bit. We're taking a quick step back.
Yeah. Trick them into compliance.
Basically.
So, Jamie, we have a biology question that came in for you.
Oh, okay.
Are there specific types of marine life expected in this area that we were
Come
hoping to
wide.
see or that we're looking for? And what are the marine biology objectives for this set of dives?
All right, great questions. So we're particularly interested in the rocky hard features because coral and sponges are the particular biology focus.
Sure. Those
One
rocks.
of our lead scientists is a coral biologist, so he's very interested in getting some coral samples so he can do genetic analyses on those and compare species distributions. But we are also generally interested in whatever biology we come across because very little has been explored in this area. So anything we see has the potential to expand known ranges of species or even be a new species. So focusing on sponges and corals, but any biology we come across is an asset, not for sampling, but also of visual interest. We're interested in the fish. So we do have some shore scientists that would like to ID the fish in the region. We're not planning to sample the fish, but getting good zooms of them is very helpful for IDing the fish in this region as well.